Hello, good evening, everyone. My name is Jiyoung Kim. I am a clinical and research postdoctoral fellow in the Department of Genomic Medicine at Johns Hopkins, and I'm working with Dr. Fahrner in our lab to study Beck-Fahrner syndrome. So today I'm going to outline several ongoing research opportunities we're currently working on with Beck-Fahrner syndrome. I have no disclosures. So listed here are our research aims. The first is to learn more about what variants cause Beck-Fahrner syndrome. So Beck-Fahrner syndrome, or TET3 deficiency, is a newly described disorder, and there is still so much we're learning about the fundamentals, including which specific variants or changes in the DNA of the gene TET3 can lead to the syndrome. The second aim is to better understand the features and the natural history of the disorder. So one of the key components to understanding any disorder is to study patients with those disorders. And so gathering as much clinical data as we can really helps us in doing this. The third aim is to better characterize the DNA methylation changes. So as discussed previously, one of the things that we've learned is that patients with Beck-Fahrner syndrome have a specific DNA methylation pattern observed in their peripheral blood. And this has helped us confirm the diagnosis in patients. Looking at DNA methylation data, we may also be able to learn more about what the specific changes in methylation mean clinically to our patients. The fourth aim is making model organoids to study the disorder at a molecular level for the development of treatments. So in order to study any disorder, it is important to be able to create model systems that we can then study in the lab to better understand what changes are occurring at a molecular level. And this can ultimately lead us to developing treatments. So in the next few slides, I will go over what participation in any of these research aims might look at. And I want to say that research is entirely voluntary, and you may decide to participate in any or all of any of the research that we have ongoing. So collecting clinical information, this allows us to understand which variants cause Beck-Fahrner syndrome. In order to do this, we collect information on the particular variant or genetic change a patient carries. In addition to this, we collect information about clinical characteristics of a patient. The information that we collect is de-identified and sent to us through the local clinician or geneticist. This means we do not collect names, dates of birth, addresses, or any other demographic information that might lead to identifying an individual. The information that we do collect is related to the medical history of a patient. So for instance, age, ethnicity, height, weight, head circumference, craniofacial features, neurodevelopmental traits, etc. We also offer the option of sending us photographs, but again, this is entirely up to the individual and their family and whether or not they would like photographs published in medical journals. So neuropsychologic testing, this is available both on a clinical and research basis. Dr. Eng has spoken about this in the previous talk, and so I'll just mention here that research in neuropsychological testing involves, again, collecting data to better understand and characterize individuals and their cognitive profiles. DNA methylation is also available on both a clinical and research basis. So clinically, it's used to help us identify which variants in TET3 are likely to be causing Beck-Fahrner syndrome. On a research basis, we want to look at this data to understand what genes are being changed, what genes have a methylation change, and what that might mean clinically to patients. And so the last part of the research involves laboratory work. This portion of the research aims to understand the underlying molecular changes to eventually find treatments. I will say that in order to be involved in this portion of the research, it does require a visit to our clinic here in Baltimore. So this portion of research does require a blood sample from the patient. We would then send this to our collaborators at the New York Stem Cell Fund. So they are an organization that are experts in turning blood cells into stem cells. They then ship us back the stem cells, which we then are able to grow into neurons or brain cells and brain organoids, which are basically just clusters of different neurons that essentially create a 3D model of the brain. By studying these neurons and the brain organoids, we're able to understand the changes that occur on a molecular level. And using this understanding, may be able to develop therapeutics for Beck-Fahrner syndrome. So these are some of the ongoing research opportunities that we have. I want to thank all the patients who have already been involved in research and who are, because patients are what really drives forward the research that occurs in medicine. And I'd also like to thank Jamie and the work she does with the Beck-Fahrner Syndrome Foundation and for hosting this event and giving us the opportunity to share with patients and their families more information regarding Beck-Fahrner syndrome and all of the ongoing research that we have. I'd also like to thank the Maryland Stem Cell Research Fund and Johns Hopkins for funding our research and allowing us to learn more about this disorder. So I will take any questions.